This is me Arzu here, and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 3 video of the development of the teeth, and in this video we will be discussing with the early bell stage. So in the previous two videos I have discussed about the bud stage and cap stage in the second video, and in first video I have discussed about the overview of the development of the teeth. So in that first video I have discussed about the primary epithelial band, dental lamina, lingual processes, successional laminas. Okay, so I have discussed all those things in the first video, and in the second video I have discussed about the bud stage and the cap stages. Okay, so there is formation of stereo reticulum in the cap stages. Okay, and there is a connections between the cell. Okay, they are just star-like star-like cells. Okay, so how they are formed star-like, okay, I have discussed about the processes there. Okay, so in this video we will be discussing with the bell stage. In bell stage also we will be discussing with the early bell stage. Okay, and advanced bell stage we will be discussing in the next video. So this bell stage, as it the many important things happen in this bell stage. Okay, so it have a larger coverage. Okay, so it have more things to be studied in this bell stage. Okay, in comparison to cap stage or the bud stage. So I have divided this in early bell stage video, and another video I will talk about the advanced bell stage. Okay, so in this video, as we are discussing with the early bell stage, so without delaying much, let's get started. So as I have written here in the bell stage, okay, so the early bell stage and advanced bell stage is there. Okay, so I will be discussing how to differentiate between those. Okay, early, how, in which stage there is early bell and in which there is a advanced bell stage. Okay, so in the last paper I have written here. Okay, so the main thing, okay, that differentiate early bell and the advanced bell is the mineralization. So in the early bell there is no mineralization. Okay, so in early bell there is no mineralization, but in advanced bell there is a mineralization, and there is also the formation of the root. Okay, so about the horse formation. Okay, Hartwig epithelial root sheath formation is taking place in this advanced bell stage. So we will be discussing with this in advanced bell video, but for now just remember this is the main thing that differentiate early bell and the advanced bell. Okay, and after that in this okay, so first line, as I have written here, after the cap stage further invagination occur that causes the formations of the bell. Okay, after that crown shape is determined here. So this is very important, that is to be remembered in early bell stage. Okay, so the most important thing of the development of the teeth that happens in the early bell stage is crown shape formation. Okay, so what is the shape of the crown is determined in this stage. So this stage is one of the very important stages of the development of the tooth. And in the next one, as I have written there is a folding of enamel organ causes different shape of the crown, which is due to the different rate of mitosis and different in the cell differentiation time. Okay, so how the different shapes of the crown are formed is due to the different rate of the mitosis. Okay, so there is a different rate of the mitosis and there is a different in cell determination time. Okay, so there are difference. Okay, so there are the difference in the rate of the mitosis. Okay, so the division of the cells, proliferation of cells is different. Okay, at different places of the enamel organ. Similarly the differentiation, okay, differentiation time. Okay, so in this differentiation time also there is a difference. So in another point, as I have written here, inner enamel epithelial cell at the future cusp tip stops dividing earlier and begins to differentiate. Okay, so at the cusp tip, okay, so if this is a okay, sorry, okay, I will do another picture. Okay, so just think, okay, just think this is a teeth. Okay, so this is a cusp tip. Okay, so at this cusp tip, this position and this position, so these are the cusp tip of the crown of the any tooth. Okay, so this is a posterior tooth or premolars or the molars. Okay, so in inner epithelial cells, okay, inner epithelial cells, that is at the future cusp tip stop division. So at the cusp tips, okay, the inner enamel epithelium, okay, inner enamel epithelium, it stops to divide and it begins to differentiate from the cusp. Okay, so the process of differentiation starts from the cusp. Okay, so that is telling in this point. After that, pressure generated from the dividing cells to the differentiating cell causes those cells to pushed out into the enamel organ in the form of the cusp tip. Okay, and cells in another future cusp area begin to differentiate, and by the similar processes result in a cusp tip formation. Okay, so what happens is there, okay, so as there is a differentiation, okay, as I have written here, the cusp of the crown, okay, the shape of the crown is formed by the different rate of the mitosis and different in a cell differentiation time. So as there is mitosis, proliferations of the cell, so those proliferating cells, okay, just understand like that, okay, so just proliferating, those proliferating cells just do pressure, okay, so they just generate the pressure, and those pressures are acted upon the differentiating cell, and the differentiating cells, okay, so the process of differentiation starts from the cusp and move towards the cervical areas. So at that point, okay, so it just pushes or the differentiating cell that would cause the, okay, so that would cause that cells to push in the enamel organ in the form of the cusp tip. Okay, so the pressure that is generating due to the proliferating cells, they just pushes the differentiating cell. Okay, so due to the more pressure generated at that area, it forms the cusp. Okay, so just I mean to say is that, okay, so just think this is a one variety of cell, and I will draw with another pen. So, and these are the another varieties of the cells. Okay, so if these cells are proliferating in number, and if this cell is not proliferating, so all those cells, okay, will just generate a pressure that would cause the moving of this differentiated cell to move upward, okay, towards upward direction, and at the tip of the cusp, okay, so it reaches the tip of the cusp and forms the cusp. Okay, so in this pattern there is formations of the cusp. Similarly, another cusp also is formed in similar pattern, and similar process result in the cusp tip formation. Okay, so similar thing, same thing I have written here. Just think, the pressures are generated by the proliferating cell, and those proliferating cell just pushes the differentiating cell towards the cuspal end. Okay, and that pushing, that pressure would cause the cusp formation. Okay, just think like that. Okay, just take this concept in such a manner. After that, I have written here cuspal slope extent and therefore cuspal height. Okay, so the cuspal slope, so this is a cuspal slope. Okay, this is a cuspal slope here, and the height is this. Okay, so this is a height from the cemento enamel junction towards the cusp tip. So this is a height of the cusp, and these are the sloping of the cusp. So the cusp slope extent and cuspal height are due to the proliferation and differentiation occurring gradually from the cusp tip to the depth of the sulcus. Okay, so there is proliferation and differentiation occurring. Okay, so due to the proliferation and the differentiation, okay, as the differentiating cell reaches the cusp tip, from there the differentiation starts towards the cervical area, and as the differentiation starts from the cusp and moves towards the cervical area, there is a formations of the slope like structures. Okay, so there is a formation of the slope like structure, and same thing I have written here. So the in another point, as I have written here, cell differentiation also proceeds cervically. Okay, and another point, differentiation of the crown shape to tooth morphogenesis is under the control of the genes. Okay, and they are signaling molecules and the growth factors. So as we know that those signaling molecules and the growth factors are responsible for this tooth morphogenesis. So the chemicals that are acting in play in this formation of the shape of the crown is the signaling molecules and the growth factors. Okay, and the cervical loop is the junction between the inner enamel epithelium and the outer enamel epithelium. Okay, so I'll show you in the picture. Okay, sorry. So as you can see in this picture, okay, so okay, so this is a picture of the early bell stage of the tooth development, and in this picture, as you can see here, this is a outer enamel epithelium, and this is the inner enamel epithelium, okay, which is columnar in shape, and this is the inner enamel epithelium that is locuboidal in shape. Now the junction between this, okay, so this is a junction between the inner enamel epithelium and the outer enamel epithelium. Okay, so this is a junction, and that junction is called cervical loop, and there is a higher mitosis. Okay, so the due to the higher mitosis here, there is a formation of the bell like structure. Okay, so the cell proliferate in this direction, and while the cells in this area are less proliferating in comparison to that in the junction, so it forms the bell like structure here. Okay, so this is a cervical loop. So sometimes they can ask you in the viva like that. Okay, now in another point, okay, so from this paper just you should have to know is that in early bell stage there is happening the crown shape determination, one thing, and another how the cusp are formed. Okay, so how the cusp are formed, as I have told, proliferating cells generates the pressure that would pushes the differentiating cell towards the upside, okay, towards the cuspal side, and due to its movement towards the upward direction, okay, there is a formation of the cusp. And as the differentiating cell reaches the cusp tip, from there differentiation starts and the differentiation moves toward the cervical areas, and that would lead to the formation of this cuspal slope and the cuspal height. Okay, so this is the concept that you should take from this paper. Okay, so now I will move to the another paper, and in another paper, as I have written here, there are 4 types of the cells that are found in this early bell stage. Okay, so as I have discussed in the cap stage, okay, there was just 3 type of cells. Okay, outer enamel epithelium was there, inner enamel epithelium was there, and also there was the stellate reticulum present there in cap stage. And in the bud stage, you would remember that, okay, so there were just 2 groups of cells, that was a peripheral cell and the central cell. So the peripheral cell was the cuboidal in shape and central cell was the polygonal in shape. Okay, I think you have remembered all those shape. Okay, so the shape of the cells are very important, they can always ask you in a viva, so just remember that. So in this now, in early bell stage, the another one type of the cell is added in this. Okay, so that is a stratum intermedium. Okay, so this is a distinguishing cell that is appearing in this early bell stage. Okay, so now there is a 4 types of the cell, so I will be discussing first with the inner enamel epithelium. So in inner enamel epithelium, cells of this epithelia, okay, so cells of this epithelium, okay, are columnar and differentiate into the ameloblast. Okay, so inner enamel epithelium means, okay, so this layer, okay, so this columnar types of cells. Okay, so those columnar cells are the inner enamel epithelium, and those inner enamel epithelial cells would differentiate into the ameloblast, and ameloblast gives the enamel. So enamel formation, amelogenesis. Okay, so how the enamel is formed is from this ameloblast. Now the dimension of this ameloblast is 4 to 5 micrometer in diameter and 40 micrometer in height. Okay, so you can remember this, but not mandatory to remember, but also you should remember this if you want to remember it. Then cell exerts organizing influence on the underlying mesenchymal cell that would form the odontoblast. Okay, so as I have told in a first video, also I have told in a second video, so this tooth development, okay, so the development is taking place by the main one thing is there, okay, that is leading to the development of the tooth, that is epithelial and mesenchymal interaction. Okay, so that is the most important point. Epithelial and mesenchymal interaction is the thing that is leading to the tooth development. So here also this inner enamel epithelium, okay, so this is a inner enamel epithelium and this is a dental papillae, as I have discussed in previous video, and this is a mesenchyme, ectomesenchyme. So the interaction between those two, okay, this layer and this papillae would lead to the formation of the tooth development, and there is an interaction that would first lead to the formation of the odontoblast in this periphery. So these structures are here, okay, so they are the odontoblast, okay, just beneath the basement membrane, as you can see or not. Okay, so I will show you clearly. So these are the odontoblast, and this is a columnar like cells, they are the inner enamel epithelium, and this is a basement membrane, and between the basement membrane and the odontoblastic layer, okay, you can see the cell free zone is present there. Okay, so I will be discussing this. This is a acellular zone, so this is very important. So there is a formation of the acellular zone between the dental papillae or the odontoblastic layer of the dental papillae and the basement membrane of the inner enamel epithelium. So there is the formation of this acellular type of layer, so you should remember this. I have written later, I think, about this, so just remember as I have written here. Inner enamel epithelium, okay, so cell exerts organizing influence on the underlying mesenchymal cell, and there is a formation of the odontoblast. Okay, so in upcoming video I will be telling there. So first of all, there is a formation of the odontoblast. Okay, so the proteins are released from the epithelium of the inner enamel epithelium. So IEE, it gives the different enamel proteins are released from this that to differentiate the mesenchymal cells into the odontoblast. Okay, so odontoblast is formed by the mesenchymal cells, and the formation of the odontoblast would again reciprocally would activate the inner enamel epithelium to form the ameloblast. Okay, so there is a reciprocal induction present there. So I will be discussing with this in upcoming videos. Okay, so stay tuned. Now after that, in stratum intermedium, so it is a distinguishing feature of this early belly stage. So there is a few layers of the squamous cell present between the stellate reticulum and the inner enamel epithelium. So this is a stratum intermedium. Okay, so there is a squamous cells. Okay, squamous like cells are present there. So I will show you in this picture. So as you can see here in this picture, okay, sorry, so as you can see here in this picture, okay, so in this picture, as you can see here, this is structure. Okay, so they are the squamous layer that is just above the inner enamel epithelial cell. So those cell layer that are of 2 to 3 layers. Okay, so there is presence of just 2 to 3 or 3 to 4 layers of the squamous cell is present there, and that layers of cell is called the stratum intermedium, and it is closely attached by the desmosome and the gap junction. So I have forgot to say you that. Okay, so in the inner enamel epithelial cell layer also, okay, so this inner enamel epithelium cells also, they are just attached to the basement membrane by the hemidesmosome and attached with each other by the desmosome. Okay, so there is a perfect connections between the inner enamel epithelial cells too, and this inner enamel epithelial cells are also connected to the stratum intermedium. So there is connection between all those cells. Okay, I mean to say is that, so this stratum intermedium is connected with this stellate reticulum also and inner enamel epithelium also by the different kinds of the gap junctions. Okay, there is gap junctions and desmosomes present there. So they are closely attached by the desmosome and the gap junction, and their well developed cytoplasmic organelles are present there. Acid polysaccharides and the glycogens are present there. So this is important to understand here. And glycogen deposits indicates the high degree of metabolic activity. So this is very important point you should remember from this stratum intermedium, that it is metabolically active type of cells. Okay, so it constitute the developed cytoplasmic organelles. Similarly, acid polysaccharides are present there, and the glycogen deposits are present in those cells, which makes it metabolically active. And also there is high activity of the alkaline phosphatase. Okay, so you should remember this points. Okay, so acid polysaccharides, acid alkaline phosphatases, and glycogen and cytoplasmic organelles are the well developed here in stratum intermedium layer, and this point is very important of this topic, stratum intermedium. So you should remember this, and it works synergistically with the cells of the inner enamel epithelium as the single functional unit. Okay, so as inner enamel epithelium is acting, okay, so it is acting to form the odontoblast, okay, by epithelial and mesenchymal interaction. So in that process, as I have discussed here, okay, so in this process also, this stratum intermedium, as it is metabolically active, so it helps the or synergistically work with the inner enamel epithelium to form the odontoblast. Okay, so after the stratum intermedium, I will discuss it with the outer enamel epithelium. In outer enamel epithelium, there is a flattened to the low cubital cell present there, and it laid into the folds. Okay, between the folds, mesenchyme of the dental sac forms papillae. Okay, so this is not that dental papillae, this is another kind of papillae here, and contains the capillary loops and provides the rich nutritional supply to the enamel organ. So in outer enamel epithelium, there is, so this is a outer enamel epithelium. Okay, so this is a outer enamel epithelium, and there happens is the folding. Okay, so folding happen in this layer and in the folds. Okay, and in the folds, so in this folds, okay, so this is a space between the two folds. So in this fold there is a mesenchyme. Okay, so there comes a mesenchyme of the dental sac, and that mesenchyme contains the capillary loops, and those capillary loops contains the blood, and they are responsible for providing the nutritional supply to the enamel organ. After that, our another type of cell is a stellate reticulum, which is similar with that of the cap stage. So it expands firstly mainly by the accumulation of intercellular fluid, but later, okay, so this happens in the cap stage, and also in the some time for this in early belly stage. Okay, so there is a firstly accumulation of the intercellular fluid, and it increases in a size. Okay, stellate reticulum increases in size, but later what happens is that, okay, so the prior to the enamel formation, means the prior to the mineralization, so prior to the starting of the advanced bell stage, they collapses, reducing the distance between the centrally situated ameloblast and nutrient capillaries near the outer enamel epithelium. Means that, okay, so this layer, which is stellate reticulum layer, so it just collapse, due to which this inner enamel epithelium comes closer to the outer enamel epithelium. Okay, and this process, okay, so this process is named reversal of nutrition. Okay, so I will explain this reversal of nutrition properly here only. So what happens is that, okay, firstly I will show you, I will explain by the picture. So firstly what happens is that when there is a epithelial and mesenchymal interaction, so there is a formation of the odontoblast, and when the odontoblast is formed, and after the formation of odontoblast there is a formation of the dentin. Okay, so there starts the formation of the dentin. Okay, just think, okay, I will do the picture. Just think, this is a inner enamel epithelium, okay, and this is a odontoblast. Okay, this is a odontoblast, this is a odontoblast layer. Okay, this is odontoblast, and this is a inner enamel epithelium, and this is a cell-free zone between the two layers. So what happens is that this odontoblast, when it is formed, they would form the dentin. Okay, so I will show you by the another pen. There is a formation of the dentin. Okay, so the first dentin that is formed is a mantle dentin. Okay, as I have discussed in my dentin video, so mantle dentin is formed here, and this odontoblast would move towards the pulpal side and giving the odontoblastic processes, and the dentin are laid in such a manner. Okay, so it will be like this. Okay, after some time when all the dentins are like, so this is a inner enamel epithelium layer, and here it comes the odontoblast. Okay, so odontoblastic layer, it comes here, and here is the odontoblastic processes, and here is the dentin. Okay, so dentin is formed here. So I mean to say is that, so this odontoblast just form the dentin and moves toward the pulpal side. Okay, giving the odontoblastic processes in between the both the layer. Okay, so what happens is that after the formation of dentin, the inner enamel epithelium, which are just differentiating into the ameloblast, so as they are doing the amelogenesis, formation of the enamel, they require the proper nutrient, but due to the formation of dentin, which due to the formation of dentin, okay, so there is cut off in the nutrition supply by the dental papillae to the inner enamel epithelium. As previously, this inner enamel epithelium was taking nutrient from the dental papillae, but due to the formation of the dentin, there is a blocking of the nutrient supply from this inner enamel, from this dental papillae to the inner enamel epithelial cell. So what happens is that at that time stellate reticulum just collapse. Okay, and this inner enamel epithelium is brought closer to the outer enamel epithelium. Okay, and there is happening also a folding in this outer enamel epithelium and the mesenchyme. Okay, so the mesenchyme would comes in that fold. Okay, so this is outer enamel epithelium, so this is outside and this is inside. Okay, so this is stellate reticulum here. So okay, so these two are different picture. Okay, so in this side there is stellate reticulum which is collapsing, and this side there is a mesenchyme. So due to the folding of outer enamel epithelium, so mesenchyme would come closer to the inner enamel epithelium, and also the by the collapsing of the stellate reticulum, inner enamel epithelium is reaching towards the this outer enamel epithelium. So inner enamel epithelium, okay, so this is a inner enamel epithelium. Okay, so this is a inner enamel epithelium. Okay, so it is reaching closer to the mesenchymal layer, and in the mesenchyme there is a sufficient capillary loops. Okay, so those capillary loops just provide the nutrient to those inner enamel epithelial cells. Okay, so this process is called reversal of nutrition. Okay, and this is one of the most distinguishing and most important feature of this early bell stage, and this feature in early bell stage make this stage as a critical stage in a tooth development. Okay, so if there is not happening this reversal of nutrient, enamel organ would have been working nothing. Okay, so it would have been died. Okay, so this reversal of nutrition is one of the most important thing that is happening in tooth development, due to which this early bell stage is also one of the critical stage in the tooth development. Okay, so sometimes they can ask you in a viva which is a critical stage in a tooth development, so at that time you should tell that. So I am discussing with every critical concept in very critical way. Okay, so I am just telling small thing in repeating all those small things many times so that you can remember, as I have told in my previous videos too. Okay, so I want you to remember all those things when I am telling you. Okay, you shouldn't have to study after watching this video. Okay, when you watch this video, you just grab all those knowledge that I am telling. Okay, so it is easy to remember all those things that are pronounced by the another. Okay, so if someone just tell you, then you will remember that thing for the longer time. But if you want to study, if you just mug off all those things, then the memory is not of long term. So just when I am telling all those things, just try to understand and remember all those points. Okay, as I am repeating a simple thing in many times. Okay, so and also dissecting all those critical concepts. Okay, so those concepts are very important, just like this reversal of nutrition. So this is also a very important concept here. Always they ask in a viva, they can see you write about the short note in the reversal of the nutrition. Okay, so you should tell all those points. Okay, so it happens in the early belly stage, and there is a collapsing of this stellate reticulum, and what happens. Okay, so everything you should have to tell there. Okay, so inner enamel epithelium, as it gets the nutrition from the dental papillae, but with the formation of dentin, there is a blocking of the nutrition, so it get closer to the outer enamel epithelium by the collapsing, and from the mesenchyme it takes the nutrition. Okay, so you should remember all those points. After that, in another paper, as I have written here about the dental lamina. Okay, so the dental lamina, it is partially degenerated, and also there is a presence of the remnant of epithelium that is called cell rest of Ceres. So I have discussed with this in a first video. Okay, after that, in the dental papillae, there is a cellular zone separates the basement membrane and the condensed ectomesenchyme, and only the connective tissue is present there. Okay, so it means that this layer. Okay, so this a cellular zone that is present between the inner enamel epithelium and this odontoblastic layer of the dental papillae is called the ocellular zone. Okay, and we just contain the connective tissue only. So after that, those peripheral cells of the mesenchyme differentiate into the odontoblast. Okay, so those just differentiate into the odontoblast. It ultimately give rise to the dental pulp, and dentin formation begins at the cuspal tips. After that, basement membrane just prior to the dentin formation is called the membrana preformativa. Okay, so this is one of the most important MCQ question. They can also ask in a viva. So the basement membrane basement membrane. Okay, so the name of the basement membrane just prior to the dentin formation is called membrana preformativa. Okay, so at this time the basement membrane is slightly more permeable. Okay, so there is a degradation of the basement membrane, due to is the enamel proteins. Okay, so it comes from the inner enamel epithelium in the dental papillae, and that would differentiate the cells of the mesenchyme into the odontoblast. Okay, so we will be discussing with this in the later video, epithelial and mesenchymal interaction. Okay, so for now, just you remember membrana preformativa. Okay, after that, the dental sac, it shows the circular arrangement of its fiber and resembles a capsular structure. Okay, and more fibrous with a 3-layer inner cellular, outer fibrous, and middle loose connective tissue. Okay, so there is a formation of the 3-layer as in the cap stage, as in the bud stage. Okay, so with the development of the tooth, okay, as it proceeding forward, there is also a development in this dental sac. It is becoming more fibrous, so that there is formation of the 3 layers, particularly inner is cellular and outer is a connective tissue layer, and in middle there is a fibrous layer. So the further development, fiber differentiate into the periodontal fiber. So this is important to remember here, as I have discussed in the first video. So enamel organ just give the enamel, dental papillae give the dentin and the pulp, and this dental follicle or the dental sac would give the cementum, periodontal ligament, alveolar bone. Okay, so I have discussed with this point in the first video, so you just watch all those videos so that you can easily relate all those things. And after that, as I have written here, it is called the critical stage of the tooth development, because the tooth shape is determined here. Okay, so this is one of the most important thing that is happening in this early belly stage. After that, identity and the functions of the cell is established. Okay, so there is also the identity and the functions of the cell is established in this stage of the tooth development. And similarly, the foundation of the hard tissue formation. Okay, so there is a foundation form. Okay, as like in the dentin or in the enamel, first of all there is a formation of the matrix. Okay, so enamel matrix or the dentinal matrix is formed. After that, there is happening the mineralization upon that So this is also forming a hard tissue formation foundation. Okay, so it is forming the foundation of those hard tissue formation, okay, upon which there is a hard tissue forming by the mineralization. So there is foundation formation. And also the another point, that is reversal of nutrition, which I have discussed earlier. So just remember all those 4 points. Tooth shape is determined here, reversal of nutrition, foundation of hard tissue formation, and identity and the functions of the cell is established in this layer. After that, as I have discussed previously, early bale no mineralization, advanced bale mineralization, and the root formation is there, and successional lamina formation also takes place in the early bale stage. Okay, and another thing I want to add here, okay, so about the morpho differentiation. Okay, so morpho differentiation and the histo differentiation, as I have discussed in the earlier videos. Okay, so in the bud stage there was just a morpho differentiation and not the histo differentiation, and in the cap stage there was both morpho differentiation and the histo differentiation, and also in this early bale both morpho differentiation is also present there and histo differentiation is also present there. Okay, so after seeing this much, okay, so I think you have also been familiar with this picture, so I will just recite once. So this is a remnant of the dental lamina. Okay, so this is a remnant of the dental lamina, that is called cell rest of Ceres. After that, this is a stellate reticulum here, okay, star like, and in between there is a intercellular fluid. Okay, water intercellular fluid is present there, which is giving cushion like consistency to this stellate reticulum, and it acts as a shock absorber. Similarly, after that there is a stellated stratum intermedium, okay, which is a 2 to 3 layers of squamous cells. So they are the stratum intermedium, and they are metabolically very active due to the presence of the high activity of alkaline phosphatase, glycogen deposit, and acid phosphatase. Okay, so acid phosphates are present there, as I have written here. So just remember all those points. Okay, so sorry, acid phosphate, no, acid polysaccharides are present there, glycogen deposit and alkaline phosphatase is present in this stratum intermedium. After that there is a inner enamel epithelial cell, which is similar with that of the cap stage, and this is the outer enamel epithelium. Okay, after that there is a cell free zone or cellular zone is there, and after that there is a odontoblastic zone present there. And after the formation of odontoblast, there is a simultaneous formation of the ameloblast. Okay, so there is a reciprocal induction between this odontoblast and ameloblast, and this is a dental papillae here. So this is cervical loop. Okay, so junction between the two inner enamel epithelium and outer enamel epithelium. And so I have completed all the, and this is a histological picture of this early bale stage of the tooth development. Okay, so I have completed all those points that are to be discussed in this early bale stage. So I guarantee you, after watching or after knowing all those points, okay, so I have taken all those points from the Orbans. Okay, so Orbans oral histology and development. Okay, so I have taken from that book, I have taken all those important points. Okay, I have prepared the proper note from the text of that book, so you shouldn't have to be worried anymore about the things that are present in this early bale stage if you have watched this video properly. Okay, so after seeing this much, I will catch you in the next video, in the part 4 video of this development of the tooth. Till then, stay happy, stay healthy, and stay tuned. And also, if you love this video, please do like, subscribe, and share with your batchmate, because making all those videos takes lots of time. Okay, it takes your lots of efforts and lots of time, so I just want your support. So by saying this much, tata, bye bye, thank you.